In this section, I'm going to talk about techniques to cultivate and treat viruses. So in vivo means in a living organism. In vitro means in a non-living organism, or in cell culture or that type of stuff, or tissue culture. So in vivo is talking about an entire living organism, while in vitro is talking about in a petri dish in some place, whether it's in cells or in tissue culture. So in vivo methods include viral cultivation in lab animals or embryonic bird tissues, while in vitro methods include viral cultivation in cellular tissue culture. The primary purposes of growing viruses is to isolate and identify them in clinical specimens, to prepare viruses for vaccines, or do detailed research. The first one, isolate and identify viruses in clinical setting, is pretty rare. We do a lot of molecular diagnostics when it comes to the need to diagnose viral infections. So just to grow them, to identify them in a lab, it's pretty rare. But preparing viruses for vaccines and doing research on them, that's pretty common. So using live animal inoculation. So remember, viruses need a host in order to replicate. So you need to actually get the virus into a living cell to make it replicate. You can inject viruses into mice, rats, hamsters, guinea pigs, into their brain, blood, muscle, body cavity, skin, or foot pads in order to get this virus to grow. Once it grows in high enough quantities, then you can isolate it from the animal. This will kill the animal. Bird embryos are also used. So the benefits is embryonic development occurs in a protective shell. So you kind of contain that virus inside of it so that it doesn't go out into the environment. There's different places where you would inject it into this egg, and you can inject it using a method called candling. So if you've ever lived on a farm with chickens that are kind of free-range chickens, and the chickens are allowed near hens, there's a potential that you're going to pick up an egg and it's a fertilized egg. So what I used to do when I lived on my farm is I would pick up the egg and hold a flashlight underneath it and form a seal around the flashlight and the egg. And when you do that, the light is forced to go through the eggshell, and you can see right through the eggshell. And you can look inside to see if you see an embryo. If you do see an embryo, you would incubate it instead of eat it, because obviously you don't want to eat a chicken embryo, or maybe you do, that's okay. But that's just one way to determine whether the egg is fertilized or not. So scientists can do that. They have a special candling type apparatus, and they can specifically inject viruses into a certain part of the egg in order to grow it. The egg does have to be fertilized, and the bird does die. Cell culture or tissue culture, this is in vitro. It's a simple and effective way to grow populations of isolated animal cells in a sterile dish or bottle, and then viruses are propagated throughout the entire cell culture. Here you can see normal cell culture, and here you can see how the virus has killed all of these cells. So that's where the virus is. Anywhere that's clear, that's where you'd find a high concentration of the virus. And those are known as plaques. So clear, well-defined patches in the cell sheet. That's where you can see the virus has changed the shape of the cell or killed the cell, and that's where the virus is. Viruses in human health, so it's impossible to measure the number of viral infections worldwide. The most common cause of acute infections that do not result in hospitalization include colds, chickenpox, influenza, herpes, warts. Some have high mortality rates like rabies and Ebola. Others lead to long-term disability like polio and neonatal rubella. All of these different types of viral infections we will talk about in more detail toward the end of the semester. So, why don't we have a lot of drugs that target viruses? Well, they mutate at a rapid rate. They're difficult to design therapies against the virus. In order to develop a therapy against something, to deactivate a virus, or even to treat a bacterial infection, you need to have a target. What can our weapon specifically target on the virus that's not going to be the same in a human? So we have to look at the virus and try to find something that's unique on the virus that a human wouldn't have. Well, humans have DNA and RNA, so we can't necessarily target their genetic material. Although now there's newer technologies where we have CRISPR-Cas9 system that can identify specific sequences and destroy DNA of specific sequences, although the safety and efficacy of that to date is unknown. But when we try to target a virus, we try to find something like a protein or some component of that virus that we can stop to either prevent it from replicating, prevent it from entering our cells, or prevent it from exiting our cells. So at least we can slow down the rate of progression of this virus. Interferon is a naturally occurring human protein that has had some success in preventing and treating viral infections, but even that is unsafe in high levels, and you have to really monitor how many doses you give to a patient. So one of the reasons why it's so difficult to develop therapies against viruses is the fact that they have a high mutation rate. So viruses are constantly mutating. They mutate at a rapid rate, and that prevents us from being able to specifically target a component of that virus. This is why to date we still don't have a cure for HIV. We've got a lot of treatments that have shown some success, but there is no cure. And it's all due to the high mutation rate of these viruses.